Hello. Marlene, let me know when we can start.
Okay, we're still waiting for a few more people to join, then we'll begin.
Okay.
Yeah. I think sorry, let me just put my video on. Hi, everyone. Thank you for joining in. And my name is Marlene Koske. I am a third year medical student at Moi University. I am also part of the organizing committee that wrote this virtual GA together. So as we wait for more people to join, I think we can, I think we can log into Mentimeter just for a bit of context of what this session is for. So just go to menti.com. I will send the text that in the chat box. I will share my screen so that we can answer a few questions and get to know each other a bit. Okay. I hope you have logged into menti.com. The code is, sorry, let me just shift this. The code is 3506897. I think you can see it on my screen. Yeah, so describe your year in a word. It's been a tough year, very different from what most of us have ever experienced, with the COVID pandemic, lockdowns in almost all countries worldwide, uh, closure of schools and most businesses. We've all gone through a lot, and I'm sure we've all had different experiences. So just let us know how your year has been in one word. It could be anything. Ah, tiresome. That's very true. And not just tired, physically being tired, even mental tiredness. That's true for me too. Okay. Yes, definitely unique. Unpredictable. That's true as well. For those who are joining in, we are at menti.com. The code is 3506897. I hope you can see it on my screen. We are describing how our year has been. And it's been different for everyone. So just let us know how you have been this year. True, it's been very long. I think that was one of the first words that came up in my mind. It has been a very long year. Also discouraging. I'm sure all of us have lost something in a way. Some of us have lost our loved ones or friends. Some of us have lost time in school. We've all lost a lot, and that can be discouraging, which is also true for me. True. Yes, it's also been educative. We've learned many different things, especially with the changes in the education systems and most of the systems in the world, from being physical to virtual. For example, this GA, we're learning how to do things differently from what we're used to. And though it is from a somewhat bad experience, I feel like the fruits are something to remember, like the education, the things that we have learned. Okay. Kuna mtu amesema imeban. I don't know what that means exactly. Maybe you can text it in the chat box. Also with what you guys have learned, especially the person who said that the year has been educative. What have you been educated on? Okay, I think I'll give this a few more seconds, this page, and then we'll answer the next question, and then we can move on to the session itself. Okay, our next question is actually about the session that we have today. What do you understand by emerging and re-emerging diseases? What do you understand by emerging and re-emerging diseases? I must admit, the first time I saw this topic, I wasn't really sure. I had never actually thought about it before. But after having researched it and looking it up, it's actually something that should have more attention because it's something that affects us directly, especially as doctors in training or professionals or just anyone. Everyone is affected by this. So, what do you understand by emerging and re-emerging diseases? Of course, that's without going to Google. Okay. I am not seeing any answers. Is this a new topic for everyone? I'm sure at least one person has some sparse idea of what it is. Just write what you think it is. You don't have to be sure. Okay. Someone has said new diseases coming up or former disease that are rising up again, which is true. Then there's someone who has said emerging diseases are those that have just come up. Re-emerging diseases are those that were previously there. Then they disappeared and now they are back. Okay. I don't know what I think about that, but we'll see what we are told during the session. What about examples of some of the emerging or re-emerging diseases that you think are there currently? Okay. Emerging diseases that are those, I think there are diseases that are novel with regards to symptomatology as well as approach to management, etc. Re-emerging diseases are diseases that have affected humankind for a while, being managed but making a comeback. Okay. So you guys actually know more than I did before I looked it up. We have someone from the chat box. Babu Owen has said that emerging diseases are new diseases, while re-emerging diseases are former diseases that are now becoming a threat. Okay, that's good. I think we all have the same idea of what this is, and I'm glad that we have this knowledge before learning, meaning by the time we are being taught or by the time we're done with this session, we'll have actually either confirmed what we already knew or unlearned what we did and actually gotten the right things. So I think we can now close this. Thank you for all those who have participated. I'm happy you guys are here and I look forward to learning with you guys. I will now stop sharing my screen. Okay, again, hi everyone. I'm sure there are people who have just joined in. As I had said before, my name is Malin Koskei. I am a third year medical student at Moi University. I'm also part of the organizing committee that planned this virtual General Assembly. And I'm happy that we have this many people who are interested in learning about all these topics that we've put together. And I hope that you get what you expected to get from it. So having finished with the introductions and the getting to know each other, I think we can now move on to the actual session. And before I allow the speaker to speak, I am going to introduce him. Our speaker today is from the University of Nairobi. He is a lecturer in the School of Medicine and he is a virologist by profession. He is an avid researcher. I confess I looked him up and he has actually published quite a lot of work. And I think, he has actually published a paper on emerging infectious diseases and I'm very impressed by him. And I think he is currently working on something to do with emerging and re-emerging diseases in Nairobi. He is Dr. Moses Masika from the University of Nairobi, and I welcome you. Karibu sana.
Hey, thank you, Marlene, for the colorful introduction. I tend towards duller ones, but I appreciate the effort you made to stalk me. The topic today is interesting, and I know some of you are expecting that we are going to have a lecture-like approach, which I don't want to take today, even though that is my source of my daily bread. I would like to have an engaging lecture to provoke your, an engaging session to provoke your thoughts and also hopefully get some thoughts from you on emerging and re-emerging infections. Today, in fact, I'm just going to focus on emerging infections without necessarily focusing too much on splitting hairs between emerging and re-emerging because characteristics tend to be similar. I spend most of my time doing research. I'm based at the School of Medicine, University of Nairobi, and also run a research lab called CAVI, Institute of Clinical Research. My interests are currently on viruses, although at CAVI we also do clinical trials for vaccines, but my interest is mainly emerging and emerging viruses. In fact, my PhD is on the same emerging and emerging viral infections in Taita Taveta and Nairobi counties. I'm going to share I'm going to share my screen now. I can see something strange here. I don't know if Marlene, you're still sharing, but let me share my PowerPoint. Then we can pick it up from there. Who can share? Only host who can start sharing when someone else is sharing. Okay. So Marlene, for some reason, the settings seem to have changed. You can see if I'm able to share my work.
Okay, let me check. I think Nick can help with that. I stopped sharing mine too.
Could you try now?
Okay. I keep getting something on advanced sharing options.
Okay.
Which I have not seen in my short Zoom life. Let me just switch off my phone. I can see one of my bosses is calling. I don't want to risk getting work at this point. Okay.
Okay.
So
And now can you try now?
who can share? I'm seeing who can share only host. Let me just put only host and see if it makes sense. Oh, all panelists. Then we can start sharing only host. Okay, so it's now agreed to share. So the topic today is mainly going to focus on historical aspects of emerging infections in a view of forming your minds and ideas to what could happen, what has happened and what could happen. And as some people suggested in the list, let me actually pop down the list of participants. I can see only the panelists and 15 attendees, but I can see some familiar names who have probably taught before. I hope you'll be active today. I'm not going to speak alone, but in terms of emerging infections, when we talk about emerging infections strictly, we are thinking of infections whose incidence has increased over the last 20 years. And there's potential for this to increase in the near future. So there are several, of course, of this. But this topic or this title of emerging infections, though it has been in literature for a while, really got fashionable in the 80s. In the 80s, when many of you were just ideas, but HIV was ravaging or just coming up in the world. And what happened in the 80s is that in the early 80s, HIV was recognized as an infection. It had been there for a while. Some people think it started in the 20s in the Congo Basin. And with time, it caught up and infected many more people to spread across the world through different routes, of course. And in the 80s, it came out. So that's when it emerged. And after that, we've had several others. We shall see some of the examples. But some of you also discussed the issue of re-emerging. But today I want to combine all of them and say that when you're thinking of emerging infections, you also want to think about infections that had probably been controlled initially and then researched. So this is not necessarily newly emerging infection, but it's still emerging because in some places it is emerging as a new, in a new geographical area as a new infection there. For instance, when the so-called conquistadors or conquerors went from Europe to South America and even the parts of America, they had illnesses that were with them for years, some hundreds of thousands of years, measles, smallpox. But to the South Americans or to the Native Americans, they were not normal diseases. These were new emerging infections. And that caused deaths in the millions. So that is a new geographical area where a disease occurs. We've seen geographical emergence in your lifetime with Zika. Zika initially being actually a forest in Kampala or in Entebbe today, but where an infection caused by Zika virus was detected or recognized in the late 40s. And later we know 5-6 years ago, Zika was the buzzword, especially in Brazil and a few other countries. And really it had gone full circle from Uganda to parts of the Indian Ocean islands to the Pacific Ocean islands, all the way to the US because of the world travel. And that's not the only geographical emergence, there are many others. We could also have a disease occurring in new populations. And we had this with conditions that are caused by immunosuppression, where people who, for example, are known to be immunosuppressed by cancer, by immunosuppressive agents, get some conditions that that they're predisposed to. Then you have a new infection like HIV that causes immunosuppression in young people, like the young males in Philadelphia and New York who turned up with the Kaposi sarcoma and the pneumocystis pneumonia because of HIV. So that was a reemergence actually of these conditions in a population that was not expected, because these were previously healthy young males who just happened to be men who have sex with men. But we also have issues where a disease that is known to have certain presentation then causing new manifestations. A good example again here is Zika, where previously it was not known to cause any CNS manifestations, but in Brazil we saw children who had microcephaly because of Zika, and this was later traced to be due to a mutation in one of the genes in the virus that made the virus more likely to attack neurons. So these are all emerging for today. If you look at diseases that have occurred since 1980, I included more than 20 years for the sake of discussion. This is actually 40 years. But you can see HIV emerging in the 80s, cryptosporidiosis around the same time. An interesting entity was what was called non-A, non-B hepatitis. And this was hepatitis C, D, and E, where people were having hepatitis, but when they used the regular test for hepatitis B and hepatitis A, they were negative. But they knew this person had hepatitis. So later in 1987, 88, and thereabouts, hepatitis C and E were in quick succession discovered or described. And so we began to characterize the non-A, non-B hepatitis. E. coli, those two strains you can see here, were also coming up. Another interesting entity is influenza. That influenza is a virus that has been around for thousands of years, but every so often it mutates. Actually, it's constantly mutating, and every so often it causes an outbreak. And once in a while, when you have a shift, which is when the influenza genes reassort or recombine to form a new strain or a new subtype, actually, the body, the human population at that point is totally unprepared for that, or only not immune at all. We have this kind of outbreaks that have occurred across the last 40 years or so. The last one being in the era of Obama presidency, which was quite a big thing. Of course, not as big as COVID today, but it was quite a big political issue, the H7N9. SARS was, and this is related to the COVID-19 you have today. SARS was in 2002, 2003, 2004, a big issue again, beginning in Guangdong in China. And because of trade that was just beginning at the time to be quite frequent travel across the world, within a few weeks, it was spreading to about 30 countries. And that is, after that, it was able to be contained. If you control, or if you compare the parallels between the first SARS and the second SARS-CoV-2, I think the second one is much more infectious, because the first one, although it was able to go across the world, did not infect as many people, but it was more lethal. It killed more people in terms of proportions. I think about 10%. About 10% of all people who had SARS died, as compared to now about 2 to 4% for COVID-19. And it could actually be less because there are many people with COVID-19 who are not counted in these numbers. Hantavirus is an interesting one. We may be able to talk about it later if there's time. And Zika, we've already mentioned some of the important aspects of Zika. The other illnesses I wanted to, or infections I wanted to bring up is, there are those that have reemerged. West Nile has been around for years, but a few years, within this period it emerged in the US. Marburg has also been around since, for decades, but once in a while it reemerges in new places where it was not there before. But you remember in Kenya, we had the first case in the early 80s where a tourist went into the Mount Elgon caves and later was treated at Nairobi hospital, dying. And one of the doctors was infected, but he survived. I'm still looking for him because I need to get his blood and probably test for antibodies in some of my assays. I think it was called some MSAKE, not the pediatrician that many of the UN students might know though. So, and there's a book about it, so it's not anything about patient privacy. I know that's a dicey issue right now. Monkeypox is one of the pox viruses related to smallpox. And in one of the labs in the US, they had this virus in the bag, in the archives. And in terms of the lab handling, some of the people got sick, and so that they infected a few people around them, and that was considered to be a re-emerging infection. So there are still labs now, like a vector lab in Russia, a few labs across the world that still keep smallpox because of the importance of it. We probably would need stocks if there was if it was to come back to make the vaccine, so that if people are careless in these labs, anything can happen, of course. But people take care not to release such viruses by mistake. We can talk about chikungunya, cholera, yellow fever, dengue, listeriosis, Ebola, but I want to move on. This is just to highlight that there are many viruses, I think over 40 of them that qualify to be emerging or re-emerging today. One of the things we should talk about is drug-resistant bugs. This could be bacteria, this could be viruses, this could be malaria or parasites. I remember when we were growing up in the 90s, we could just go to the counter, over-the-counter shops in the village and get drugs for malaria. This, I think, contained chloroquine or fancida. I'm not so sure what they were, but they used to work. I don't even know if it was malaria fever we were treating, or it was just a viral infection, but sometimes you could be quite sick and you take a tablet and it works. But we came to pay for that later because in the notice in the 2000s, we noted that the drugs we're using were not working very well, and progressively we had to change to artemisinin and other artemisinin-containing treatments because of drug resistance. This occurs much more often with bacteria. It also occurs with viruses, especially those that have treatment like HIV, hepatitis B, herpes, hepatitis C, and all that. And every time you have a new resistant bacteria or organism, then that is an emerging infection, because then it requires a new approach. Now, this is useful for us to note. This is one of the charts I use frequently because it shows us the interaction between us and the environment between us. Many times as doctors, we often consider a lot of the other cadres in the healthcare profession as peripheral, and it's really unfortunate. I think our upbringing in medical school may have something to do with it. But as you interact with others in the space, you get to realize that as a doctor, you play a key role, but that would be totally ineffective if the others didn't do their job. And one of the approaches that embraces a unified management of health challenges is this One Health. And this One Health approach has allowed us to bring together environmentalists, bring together medical doctors, epidemiologists, bring together veterinarians and other mammalogists to counter or to try and control emerging infections. This is because when an infection occurs, it most often starts with circulation in animals. So for instance, COVID-19 may have been circulating somewhere. Say, for example, we do not have complete evidence where this was. Some people have mentioned bats. So say it was in some bats somewhere. And with time, it keeps circulating, and with time, it crosses over to other animals. Maybe because a snake eats bats or some other animal that interacts closely with bats. And then it crosses over to other animals around that are susceptible. And with time, because these animals are closer to humans, then there's a spillover to humans. And these spillovers are many times abortive, that they don't cause infection. Because for a virus to cause infection, it needs to be it needs to have the right surface antigens to infect human cells. And this often is not the case. So many times there'll be a spillover, it's limited. Maybe one person is infected because there was a high viral load in the infection inoculum, but they're not able to infect other people efficiently. But with time, the viruses mutate. The viruses are mutating throughout. And by chance, some of these viruses become or acquire attributes that make them very effective in infecting other humans. And then you get amplification within the human population, causing an outbreak. Some of them will continue circulating in the animal population without causing an outbreak, especially if they don't spill over to the rest of the population. but these opportunities for spillover are quite a number as human and animal interactions occur. Like, for example, when people are constructing roads, or when people are hunting, or people are even eating these animals, as well as displacement. If you have a national park like Nairobi, and you want to build a road or a railway across it, you're going to displace animals. These animals will be moving to other areas. They become internally displaced animals. And with time, they will meet with other animals where they go. For example, we have refugees coming into Nairobi, into Kenya, they will meet with other Kenyans there. And these new populations, even within the park that rarely interact, will now be interacting more often. And we may have a similar situation as we had in South America when the conquistadors came and brought new infections to a vulnerable population, because these animals are interacting with animals that they may not have interacted before, and some of them could be vulnerable to the infections in the other group, and that causes an amplification of an infection. And if there's an opportunity, this could also affect humans. So we need to appreciate the role that the environment, the climate, and also other personnel, not just medical doctors, but other personnel, be they veterinarians, be they mammalogists, be they ecologists, be they epidemiologists, the role they play, because all these are important in controlling such infections. And I made this in bold so that we appreciate that a majority of the emerging infections are zoonotic. They emerge from animals. And these are viruses, for example, because most of them are viruses that will have the ability to infect more than one host. They are able to, so think of viruses that can infect more than one host. Corona is a good example. If you are thinking of measles, it's difficult. Actually, measles has been around maybe for 10,000 years. It's a shame to us as humankind, we will not be able to eradicate it because we are the only hosts. Why can't we eradicate it? But if you look at viruses like corona, viruses like Bunya viruses like Rift Valley fever, these are viruses, or even rotaviruses. These are viruses that have a broad host range, and so will thrive and will keep emerging and re-emerging. And a good example is rotaviruses, where kids are given the vaccine for rotavirus diarrhea, but they still get diarrhea even though it's mild, because this virus is mutating and shifting all the time, causing the occurrence of new strains that the vaccine is not effective against. So what do we need for a pathogen to emerge? One is a pathogen, of course, that can infect humans. You could have, for example, there was a while back, I remember we had an outcry about feline HIV or feline immunodeficiency virus. I may not call it HIV. And I don't know who tested it, but there are people who are talking about HIV in quotes in cats. But this is a virus that cannot infect humans because there is host restriction. So you could find somebody interacting with a cat, they scratch or they bite, but they cannot get HIV from the cat because this cat has a different virus called feline immunodeficiency virus that cannot be crossed over to humans easily because of the host restriction. So if a pathogen is there and it cannot infect humans, it's a dead end. It will not, even if it crosses, even if there is inoculation, it will not cause an infection. It will not substantiate. But we also have pathogens that can infect humans, and on top of that, they can be transmitted across humans. There are pathogens also after infecting humans, even influenza viruses sometimes. You hear of highly pathogenic influenza viruses that are only able to infect from chicken to animals, to humans. But after that, they're not able to go from human to human. And that was the story with COVID-19 initially, that people thought it was not able to cross between humans, it was just from animals to humans. But with time we realized we were wrong, that COVID can actually be transmitted between humans. And this is what causes amplification of the infection. Between humans move much further than animals. And because of that, a cow is not likely to take a flight from China to Canada unless it's in a can of sorts or in a meat pack. So that transmission between humans is the source of amplification for any infection. But more importantly, you also need a vulnerable population. This has occurred very well with the measles. There are people in the US who have quite strong anti-vaccine campaigns, where people come up with very convincing ideas and stories and very loud campaigns against vaccines. And some people get convinced not to do vaccination. And with time, because they're creating a clout, with time, a significant number of people don't take these vaccines. And if one person, for instance, gets infected with measles, then it's easy for measles to re-emerge in that community because there's a large population of people which is vulnerable. And that is the same thing with many other infections. Like if smallpox today was to come back, most of us would get it and many of us would die because we are vulnerable to it. We are no longer getting the smallpox vaccine. I'm not saying we should start getting it, we should just make sure it doesn't come back. So vulnerable populations are key. And this is the story that makes the campaigners of herd immunity for COVID-19, that is a very strenuous theory, that you can let COVID-19 die out naturally without a vaccine, for example, without taking measures, because everybody is vulnerable, except now maybe about 10 to 20% in different populations that have been infected. And even for those who do not know for how long they will have that immunity. So most of us are still vulnerable. It's unlikely that we will get to 70% or 60%, which is what would give us herd immunity, without losing an unacceptably high number of us to the virus. What drives? So once you have a pathogen, once you have a vulnerable population, what will drive the emergence? What will fan the flames of emergence? So one is human factors. Human factors think of things like trade, things like how we behave around animals. So if you have enabling human factors that allow interactions, then you're going to have an outbreak span. One of the things, some of you remember history, I'm one of the people who like history, is in what was called the Neolithic age, or the first agrarian revolution, where basically people started cultivating farms and being able to cultivate or get enough food so that they could live in larger groups, eventually in cities. This became actually when diseases started to emerge. This is when we think things like smallpox and measles began. And of course, this happens slowly. But the fact that you have humans aggregating in one place, then that gives a disease an easier target. It's, for example, if you have a city where the average household size is 5 people, as compared to another city where the average household size is 2 people, then the city with more people per household, even if the population is the same, is the city with more people per household to have more risk or higher risk of transmitting disease. So this is a human factor. I hope my internet is still there. A human factor that can contribute to the speed of emergence. There are also pathogen factors, and we've seen pathogen evolution. One of my best examples is chikungunya, which was limited to East Africa until 2003, 4, 5, and then it mutated after an outbreak in Lamu to be able to expand its vector range. Initially, it was just being transmitted by Aedes aegypti. After mutation, it was able to be transmitted by Aedes albopictus. Now, Aedes albopictus has 2 major characteristics that make it a very good vector. One is that it's highly anthropogenic. For those of you who skived entomology classes, that means it feeds more on men than on animals. We know mosquitoes have different appetites. There are those who prefer to feed on snakes, on cows, others prefer to feed on humans, and some mix. So Aedes albopictus is more anthropogenic than Aedes aegypti. And additionally, it has a wider geographical spread, so across India and more temperate lands. And because of that, chikungunya is now in all the continents, maybe except Antarctica. And so the pathogen factors such as that would be useful for predicting emergence. The environmental factors like drought, famine, and also climate change. An example is some of you might have read this topic, might have seen something called the Sin Nombre virus, or the the Four Corners virus. The Four Corners are states in the US that I think Utah, Colorado, New Mexico, and Arizona, that have one corner where they all border each other. And that is a reservation for Native Americans, the largest reservation, I think, in the US. And there was, in 1992 or 1993, I think, a strange disease that emerged there. Somebody dying very quickly, a very young man dying very quickly of pulmonary syndrome or pneumonia, which was atypical. And it looked like, a normal death until some epidemiologists or medical personnel noticed that the fiance had also suffered the same. And with time, they managed to link this and find more people. And eventually they linked this to rodents. And after a few tests, discovered that it was a new form of hantavirus. Now, what had happened is that there was a surge in rodents. In the previous 2-3 years, there was drought. And then this time around, there was heavy rains that caused a flux or a lot of food. And so these rodents respond very well to availability of food. So they will give birth or they will reproduce very fast. And so because the numbers were increasing, then it was easier for them to transmit this virus, and also amongst themselves and also to human beings. So that's one environmental factor. Another one is because of global warming, which I hope all of us understand, not just as a word, but what is really happening and causing it, that we have now pathogens that say are transmitted by mosquitoes because expanding in range, because mosquitoes are now found much in much broader latitude than they were before. We have global warming causing mosquitoes to go much higher with the latitudes than they would before into the temperate and subtemperate areas. And if you have an Aedes aegypti mosquito, all you need is one person with the disease, say dengue, and then you will have many more people exposed. There was a paper, I think, in this year or a year ago in Egypt. Egypt is one of probably the leading country in Africa with, somebody has raised a hand. Go ahead, Kate, unless it is an accidental hand raise. Okay, so I'll continue. If there's anything, Marlene, let me know. So in the 1940s and 50s, they cleared all the mosquitoes using DDT, I think, which has now become probably unacceptable. But back then they did it. And for them, it was important because to date, Egypt is probably the biggest tourist site destination in Africa. Sorry for the Kenyans who thought it was Masai Mara, but Egypt seems to attract many more, probably 10 times more. So because of that, they did not have issues with dengue and what have you. But a few years ago, there was one of the tourists who went back to Italy and was diagnosed with, I think it was dengue or West Nile. And later they found that there were Aedes mosquitoes in pockets of the cities around the Red Sea. I think it was Alexandria, if I'm not wrong. Or either the Red Sea, yes, it may not be Alexandria, but with the new findings, of course, it means that is a risk for these infections to reemerge. That they were not there before, but because the vector is back, it may have come in a ship, in a tire somewhere, and now once it gets the right conditions, warmth and its eggs, and they multiply, then it's possible to have a resurgence of such infections. Sociopolitical factors are also important. Think of a country like Congo today. Congo is undergoing the DRC, that is DRC Congo, is undergoing the 10th Ebola outbreak, which is the largest in its history. It's the second largest after the West African Ebola outbreak, but it's the largest in Congo. And so there are people fighting for survival. People have been dying every day from militia activity, and then there's Ebola. So these sociopolitical factors hinder the ability of a country to respond, to detect, and to control emergence. And it's a serious issue. Bioterrorism is also real. We know people have tried to use anthrax and other infections for bioterrorism, and also technology. 50 years ago, that was what, maybe 1970, it's too soon. Maybe 1950, it would have been quite a task to travel from Nairobi to Kisumu. And so if somebody had cholera in Nairobi and they decide to go to Kisumu, the people of Kisumu will be safe because this guy will never get to Kisumu. He will die before he gets to Kisumu. But today you can be in Nairobi right now. I could be giving this talk and take dinner in Kisumu in an hour or two. So if I had cholera, I will be okay and I'll still get to Kisumu, and then I'll be able to spread it very easily. Maybe now with Zoom and these other things, the technology will start undoing because now that I'm here, I'm not likely to infect anyone in my office. So technology could also work to reverse some of these issues. Transmission is broad. So some of the things we know to transmit infections, emerging infections are arthropods, vertebrate vectors. So those two are main because most viruses have broad host range, and most emerging infections are viruses which have a broad host range, and these kind of vectors are good for their transmission. The other thing we notice, of course, the viruses tend to be RNA viruses, and I would like you to tell me why you think they are more likely to spread or to emerge than DNA viruses. And you can also get this through direct contact. So somebody with Ebola bleeding, or even the normal body fluids, if you're in close contact with them, you might get it. Vertical transmission from the mother to child, we've seen this with Zika. Sexual intercourse, interestingly, that's a very interesting concept now, because Zika, Ebola have all been shown to be transmitted mainly from male to their female counterparts, not the other way around. I'm not so sure if there's any cases of the other way around. I think there may also be man-to-man transmission, vertical sexual intercourse, and of course inhalation, which is, I would like to say, airborne transmission like we've seen with COVID now. This is, I think, 2018. Part of the work I'm doing on my emerging and re-emerging infections with some colleagues from Helsinki. These are tires in Changamwe in Mombasa. And these tires are left in the open, so when it rains, they collect water, and mosquitoes love them. So in fact, it got to a place we were just looking for mosquitoes in tires, because we were looking for the particular Aedes mosquito breed. And Aedes mosquito is what is known to transmit many viruses. And because we were hunting during the day, they tend to rest in these parts. And then in the evenings, they begin to go out, and not at night, late afternoon, in the evenings, they go out to bite people. And these tires were teeming with mosquitoes. So if you have a cousin who sells these tires, please tell them to cover them so they don't take in water and harbor such mosquitoes. It was everywhere we went with tires. Even if it was a single tire, we would find mosquitoes because there was water inside. We also know our interaction with animals, that our appetite for game meat, be it monkeys or zebra or whatever else it is in different parts of the world, contributes to our interactions with animals. And of course, before you make a meal out of such an animal, you might get some scratches and bites, and that is likely to cause, or even cuts as you prepare the meal, that might cause issues. There is also an interesting, this is Equatorial Guinea, by the way, it's not somewhere out of Africa, where you can see one of these animals called a pangolin was thought to be linked with COVID-19, but later refuted. I didn't know we had pangolins in Kenya until one of my colleagues in the Museum of Kenya was called to, told me there are so many mammalogists, and was called to respond to one that had died mysteriously. But mainly, they are not seen because most of them are traded. The scales are thought to have some mysterious powers. I can't remember what. So people kill these animals for all sorts of reasons. So I can imagine somebody thinking that they are taking pangolin soup for some mysterious superpower. But this kind of interaction with animals exposes increases our risk for exposure to different diseases. This is in Myanmar, I think, or Indonesia. Yeah, Indonesia. This is a wet market with animal meat. You can choose your delicacy. And of course, again, catching and preparing them is increased risk. And sometimes they're kept in a market where they are alive before they are killed. So you may find a snake there and a rat and other animals still alive. And infection can occur even across these animals and also across to humans. There are also, besides feed, that we kill animals for their trophies. This is pangolin skin that I was showing you before, some horns, and some scales. I can't tell what these are for. Looks like rhino horn, some skin, and what have you. And this is porcupine spines. So all these are opportunities for us to interact with animals. And this occurs even in Kenya. I'm sure you've seen the fight against hunting elephants, and it's not been won yet. So we have a lot of this in Africa, as well as Asia and other countries. So once these diseases emerge, one of the things that has been a challenge is diagnosis. And this is where I'd like to open your minds now. Some of you go to practice that you need to think about besides doing the regular tests, you need to have this open mind that when you keep seeing a similar presentation and you really can't figure out what is happening, what is causing these infections. And it may be a challenge because you don't have a very good database where you can see how many patients have come to you this week with fever. Maybe there were 100. You can't tell what is the difference between this week and last week. So when there's a surge, you may not notice. But if you're able to build such a system wherever you work or wherever you're going to be working, it might help you, save you and save the country a lot of problems. And some of the things we do, although some of them have limitations, or most of them have limitations, are things like serology. For example, today, if you tested somebody for dengue and they were positive for dengue on an antibody test, it may not necessarily be dengue because of cross-reactivity. So you know that is a flavivirus. It could be West Nile, it could be yellow fever. Maybe they even got a vaccine for yellow fever. So all this plays around in making a diagnosis. If it was negative, then you know this is something else. Same thing with alpha viruses like chikungunya. And even now with coronaviruses, you've seen a bit of cross-reactivity. So another thing you can do is and there are many ways of testing serology I don't want to get into because I said I don't want to make this very academic. Nucleic acid detection by PCR is also important. It is done for specific, mostly for specific infections. So for example, when you're doing PCR, you know, I'm going to detect HIV, I'm going to detect malaria, or whatever it is that you're looking for. But you can also tweak it if you have the capacity in the lab to detect broader categories. So you can have a PCR that detects all malaria parasites, all Plasmodium parasites. So if there's a new malaria parasite like Knowlesi, you'd be able to see it. You can also do what I did, for example, in my PhD was targeting alpha viruses and flaviviruses with what we call consensus primers. This is, for example, knowing that dengue is a flavivirus, but there are others in that group, like West Nile, yellow fever, and what have you. So you take a portion of the genome that is common to these three, but is also unique to that family of viruses, like flaviviruses. And using those primers, if you do get a positive in a sample, then you know it is positive for a flavivirus, not for dengue or for this specific virus. And then from there, you do more specific tests, like sequencing, using Sanger sequencing, which is a short, usually PCR gives you very short parts of the genome. It doesn't give you the entire genome. It gives you like 200 base pairs. It could also give you 1,000 or maximum 1,500 base pairs. But with that, you can even get to know what is the type of the virus. If it is dengue, you can say it is type 1 or type 2 using Sanger sequencing, which is very accurate. But more increasingly, we are using next generation sequencing, which is much broader. It is able to do unbiased testing. For example, in some of our samples, we just picked, we were collecting samples from bats and from rodents, and we just sequence them using next generation sequencing. And this is an unbiased form which, instead of using specific primers, use general primers. We call them random primers, which will just blast or amplify anything inside that sample. Unfortunately, you get host DNA most of the time, like 99%. So you have to use computer analysis to get the non-human or non-host genomes, and then use again the databases that are available online using bioinformatics analysis to try and find out what do these genomes belong to. And in the process, we actually discovered a new virus, although we were the second group to discover it, but no one else in the country had done it. And also in Africa, we were the second group. Actually, in the world, we were the second group to find it. Why? Because we were using, although we were not looking for it, we were using a method that could allow us to find a virus even if we don't know it. Of course, it is not that simple, but that is the idea, that you're using something that will help you detect a pathogen even if it's not what you are testing for. And that is next generation sequencing. You can also do cell cultures. The issue with cell cultures is that it's a hit or miss. For example, if you're culturing COVID, somebody was, say, culturing corona, this SARS-CoV-2 in mosquito cells or mosquito cell lines, it may not grow. Like, for example, I was able to culture dengue virus on mosquito cell lines, and I was not able to culture the same sample on viral B cells, which are human cell lines. So that is the issue with the known viruses. You don't know where they will grow. So if you have a sample, you suspect it's a new virus, you have to grow it in different cell lines, and you require a high-level security lab. If you don't know what it is, level 4 would be sufficient. And I think there are only 2 in Africa, 2 or 3. One is in Gabon, the other one must be in Senegal. So that is a limitation. And so most people don't go that route. Electron microscope is also nice. Actually, if you are looking at a sample with COVID-19 with electron microscope, you would have seen a characteristic shape of the coronavirus. And then from there, you have a clue as to what else to do to test. But these may not necessarily be available to you at your clinic, but they're available now in the country, able to do next generation sequencing, we're able to do isolation and cell cultures. We may not be able to do electron microscopy generally yet, but I'm sure with time it will be available. This is the story I was telling you about the bat. So we had some bat in Taita Taveta, somewhere near Mwatate, which had this virus called Bombali. Bombali is a district in Sierra Leone. There were scientists who were working before us in 2018 that found this virus during the aftermath of the 2014, 2015, 2016 Ebola outbreak. And so for us, it was easier because there was already an existing genome in the database to compare with and find out what this is. And after we found it, of course, what you do is now go further, do PCRs to confirm this is an RNA virus, this is a DNA virus, look at different samples and see. So for example, we detected it in mouth swabs, but we were able to find a lot of it in the lungs. After later, now once we found the virus, we went back now to look deeply. We looked at the lungs, we looked at the other organs. So when we get a bat, we usually sacrifice it and harvest these organs so that we can go and look for viruses. Additionally, we also we had collected at the same time blood from humans who lived there. And especially people would say that they had interacted with bats. We looked at their blood. Fortunately, none of them had antibodies against this virus, showing that there was no exposure. So this is a pathogen that we could not prove infects humans. We don't know if it has any ability to infect humans. Why? Because the people we screened there did not show any previous exposure to this Bombali virus. A year later, we found it in Busia. This time around, we found it in more bats, I think 2 or 3 bats. And again, screening the people around Busia. I think that's data we haven't published yet, should be coming soon. We were not able to show again that the people who had been interacted with bats in this area had been exposed to this virus. So that's a good thing. Maybe it's a new virus that doesn't infect humans yet. Remember, mutations do occur, spillovers, opportunities for spillover do occur. But that also brings a challenge that even as we do surveillance, and there are many well-funded organizations that are doing surveillance across animals to see if they can predict the next outbreak. Things like Predict, which is funded by USAID, Global Virome Project. So you have now numerous viruses that have been discovered, but the challenge is for you to tell, or to be able to tell, is this virus of any clinical significance? So that is where the research is going on now. How do we predict that Bombali Ebola virus, yes, it is in bats, it is in circulation in Kenya, it is in circulation in Sierra Leone, meaning in between Sierra Leone and Kenya, there could be numerous areas where this virus circulates, but is there an opportunity for Bombali Ebola virus to infect humans? We know about Reston Ebola virus, which doesn't cause disease in humans. It only causes disease in non-human primates. We know about Thai forest or Cote d'Ivoire Ebola virus that was only found in non-human primate, has not been known to cause any outbreaks in humans. So this could be similar. Also, it could be as bad as Bundibugyo or Zaire or Sudan Ebola virus that is known to cause significant outbreaks. Not to be hard hit by this chart. This is a phylogenetic tree. A phylogenetic tree is basically a family tree of viruses in this case. And in some of the samples that have been collecting in Taita Taveta in 2016, we are able to isolate 5 viruses that you can see here highlighted in red. And what you do is that you put this data together with existing data. other viruses that were isolated earlier, 2014, 2017. When we were doing the analysis already, the guys had collected and presented data for 2017 in Malindi. And then other strains from 2013, 2010, 2015 in India. And what we found is that the dengue that was in Taita Taveta was actually closely related to dengue that was in circulation in the coast. Taita Taveta is in the coast, yes, but it's isolated. So somebody has actually to travel for several hours from Voi or Matate or Undanyi to Malindi. But this shows you that there is urban to rural spread of dengue because of the close interaction. A lot of people in Taita will not come to Nairobi. They go to Mombasa to look for work. Some of them, of course, come to Nairobi, but many, a majority go to Mombasa. Additionally, we noticed that the virus was closely related to previous outbreak in India. And we know in Kenya, the close interaction between India and Kenya, social and business. So a lot of things, a lot of travel in between. And this has been going on for centuries, even before the British-led railway construction. So this, especially from Eastern India, the Gujarati, and also South India, in the parts that are nearest to East Africa. So this is a confirmation that this disease is global. The dengue may have not begun in India, but it has spread to India and come back. And of course, because there are strains, we can see the different strains and mutations that are sparring this infection. In fact, currently we are working with a team from South India in Bangalore to try and get or design a vaccine that would work for more strains of dengue. And this is part of the data that informs such kind of activities when you look at the variation between one episode of one outbreak and the next. And also that dengue is no longer just an urban outbreak. It's now getting into rural areas because the parts where we found this virus in Taita Taveta are quite rural. Although some of the people had traveled, it shows you that because we also found Aedes mosquito in those areas, so also trapping Aedes mosquitoes, that is a potential for a recipe for disaster, or actually an outbreak, not a disaster. It may not become sensational. The challenges also that I think I should bring to your attention, that even as we think about fighting emerging infections, there is unpredictability. Nobody could have told you in December or in January that we're going to have COVID. In fact, when China was suffering, many of us were very optimistic that it will be like SARS. A few countries will get hit, then the thing will just burn out. But that hasn't come. Been proved quite wrong, if you can be any wrong than wrong. So that is the unpredictability that these diseases present. That despite the tools we have now, we are still not able to get a model that predicts the behavior of these infections accurately. Maybe we shall, maybe we shall not, but I always believe as we get more tools in terms of satellite imaging, in terms of data collection, big data collection, and analytics, we may be able to stay ahead or at least keep up with these viruses. There are also numerous pathogens. There are hundreds of viruses that could emerge as infectious, as new infections, and that is a challenge. You don't know which one to follow, which one to study, which one to make a vaccine for, which one to make a drug for. Now, you've seen how quickly we have, the world has come up with potential vaccines for COVID-19 because of the potential for business sense. But if you have a virus like SARS, the first one in 2002, there were hardly any vaccines. Same with MERS-CoV. Why? Because there was very few people infected. And so if there are pathogens that are known to exist, but they are not known to infect people, there tends to be less research towards these things. And that is the way our resources are allocated. So we probably need to look at that and see if there's a way to refocus our energies to the most likely pathogens. But first we need to know what is the most likely pathogen, even if it's not currently infecting people. Lack of appropriate vaccines and antimicrobials has always been a challenge. If we had a vaccine for COVID-19 by now, we would have been back to work. Our challenge would have been how to get it to everybody, all healthcare workers, policemen, the old people everywhere. And then finally, guys like you who are young and energetic will get the jab. Maybe because the healthcare workers might get slightly earlier than the rest of the population, of course. And then animal reservoirs. Remember I talked about measles. That measles having no animal reservoir should have been eradicated because you have a very effective vaccine. but it's not. Why? Why is it that we're still struggling with measles? Why is it still that we are struggling with polio? Although now in Africa it's been said to be eradicated or eliminated. If we do not eliminate those, do we stand a chance at eliminating COVID-19, for example, that could have animal reservoirs? Because we cannot kill all the animals. So these are challenges that are presented by emerging infections. Diagnostics are also a challenge because it's a new virus. Like you saw how much you struggled before we could put in place any meaningful diagnostics for COVID-19. Today, we're still struggling with the antibody tests for COVID-19, which is a useful screening tool. In fact, earlier this afternoon, we were discussing one of the assays that we could use for that. And also the capacity, whether it is lab, whether it's human resource, whether it is bed capacity, whether it is political capacity, whatever form of capacity is always a challenge, and you can never have enough if you face a formidable pathogen like COVID-19, for example. So we need to think about this and keep investing until we are sure that we are ahead of virus. And to mitigate, we need political will. You've seen the effect the BBI has had, the campaigns on COVID-19. Funding, you've seen the kind of civil unrest or rather labor, industrial unrest we have in the health sector because funding has been an issue there. PPE is not enough. Even testing at some point was a challenge. Now we are talking about funding for vaccines. How do we get everybody immunized without sufficient funding or without allocating sufficient funding? Because I think these countries have enough money. We just have other reasons or that we think are better for keep taking our money away from health. And capacity building is important. You've seen the countries that have prepared vaccines quickly were not just sitting idly. Take Moderna, for instance. Somebody the other day was saying Moderna was prepared within, I don't know, a day. The Moderna vaccine is a messenger RNA vaccine. But the technology was not developed in a day. The technology to develop that vaccine in a day was developed over many years. So if we do not have that capacity now, we're going to remain behind the rest of the country. One of the things that we've been pushing for those of us in research is that the STI Act, this is Science, Technology and Innovation Act, that has 2% of GDP should go to research. And so if we got this to research, not just health, we have big challenges like food production, drought management, transportation systems, urban challenges, or urban planning. All these could count as research. But if we gave 2% of our GDP to that, we would be able to build capacity that would make solutions for emerging infections much easier to get. You will not start developing a vaccine when COVID starts and then think you'll create it in time. It takes years to develop it. The Chadox vaccine that is being being tested by Kemri is using the Chadox vector, which was developed, I think, more than 10 years ago by University of Oxford from a chimpanzee adenovirus. That's why it's called Chadox. So this technology is existing and people just tweak it. In fact, previously, Oxford had been funded to test a MSAKE vaccine on Chadox. And MSAKE is very similar to COVID. So with a very little tweaking, of course not to trivialize it, you're able to get a worthwhile product that is now a potential vaccine candidate. But if you are to start today to make a vaccine and you are competing with a virus like SARS-CoV-2, which is spreading like wildfire, then it doesn't make sense to start funding when the problem occurs. You need to do this systematically. You need to do it even when there's no problem, so that when the problem occurs, then you are ready for it. Surveillance is also important. We do surveillance some of our areas, but we only do this intermittently because, of course, we have limited cash. But if the healthcare facilities across the counties would be very good surveillance centers. For example, we'll just be picking from each facility 10, 20 samples for people with fever, 10, 20 samples for people with diarrhea, 10, 20 samples for people with meningitis, 10, 20 samples for people with upper respiratory tract infection, and pulling this together and testing them in a central lab or in several central labs. And that would be useful for telling us what is in circulation. And you can see a spike. When there's an issue, you could be able to tell from this region, we are having this problem. So surveillance is good. It can also go to the animals. It can also go to the climatic changes, because that affects the movement of animals and human beings at times, because there are still nomadic people in the country, and that would help us with tracking transmission of diseases. Vaccination, of course, when you can. Not very many viruses vaccines, emerging viruses have vaccines. And we can see we are almost getting to this stage for COVID-19. So who do you think should be immunized first? Yeah, there have been proposals that we should give it to healthcare workers, teachers, policemen, those with underlying conditions like diabetes, hypertension, obesity, and what have you, and then eventually everybody else. So those strategies will always be there, but remember there are other issues with vaccines. What is the storage temperature? How do you transport it from Nairobi to Lodwar or to Donyang or wherever these people are? How do you ensure that the viability of the vaccine is retained during transportation? And then there's research and development. And research and development for emerging infections is quite broad. There are people who look at archaeovirology, for example. You want to get a mummy and test if they're having any viruses that are related to what we have today. And that could help you show how these viruses have evolved over time. There are people who are doing real-time testing at hospitals. So you're linked up, say, with Kenyatta National Hospital, and every once in a while you pick these samples and check for epidemiology of different infections so that you can track them. But there's also a development bit where we have people testing new medical products like drugs and vaccines, diagnostics for different viruses or different infections. And all this should work together to help us mitigate against the impact of viral and other emerging infections. I would also like to highlight that you need to think about antimicrobial stewardship. As you start to prescribe, don't just prescribe antibacterials because the patient has required it, especially if you're practicing in the private sector. That is common that somebody will tell you, amoxicillin does not work for me, give me something stronger. Because the insurance company is paying, you should be wary of such. Don't just dispense because you're creating resistance where there are chance for resistant bugs to thrive. Vector control, I don't want to get too much into that, but that's also important. Information, education is key. We've seen the effect of misinformation on social media. I think this is the first outbreak we've seen the impact of social media. I think previously WhatsApp was not quite strong and Facebook and these others. The other day I saw a clip on somebody who died in a matatu and everybody was up in arms talking about COVID. Later it emerged to be myocardial infarction, I think. So without information, then, or the right information, then we are fighting a losing battle. So even in your circles, take the opportunity to educate people. Don't start spreading rumors before you confirm. Even, I mean, some stories are very sensational. And so if you are not careful, you may fall for the temptation to share the gossip or to be the one to break such information to your friends. Always counter check before you share such information. And when somebody, your friend or somebody who are in the same circles does share, try and fact check. Look at the source of that information and see if you can verify whether it is true. We also need to look at developing early warning systems. This is an ongoing activity. So a lot of work going into this, from geography, GPS, satellite imaging to epidemiology of surveillance and what have you. International health regulations. One of the issues that we keep grappling with is whether banning flights would have helped control COVID-19. The truth is that unless you lock all the flights, that no one is coming in, no one is going out, then you are just not doing anything. You just can't close the borders for one country and expect that no one will come in. They will find other routes. And we see it even at our borders. If you close the Malaba border, there are other panya routes that people use. So this is interesting in how we manage human beings. And of course, global public health surveillance networks like the influenza network, which has been very useful for COVID-19 and what have you. This is Ebola in West Africa. I didn't think I would be talking 1 hour into the seminar, but I hope you guys are still awake. Shortly, I'll give you a quiz that has no correct answer so that you can jog your minds. I don't want to tell the story about this because of time, so I'll move on to this. Now, for about 2,000 years ago, actually 2,500 years ago, there was a plague in Athens. Athens is a city in Greece that is probably one of the oldest cities that has survived till now. Of course, there are others like Baghdad and what have you. So in the 430, 27, 28 BC, there was an outbreak, a plague, and this was written about by several historians. One guy who actually survived it, and I hope Africa can begin to document some of our history like the Greeks did and the Romans, because it's usually quite good learning points. And with this plague in a city of about 400, 500,000 people at the time, a quarter died. Now, usually it was not that populated, but Athens was at war with Sparta and the allies. And so because Athens was known for its navy power, for its maritime power, everybody came to the city because they could retreat into the city and protect the waters around it. So that means people from across the area that was under Athens' control came into the city, crowding it. And of course, when there's crowding, there are issues with sanitation, issues with population control. And then soldiers at the time were also coming in from places they had gone, like including as far as parts of Iraq and Iran, where there were other wars to fight, and then now coming in back to their city. And others had actually come down south to parts of Africa. And they were also trading with Ethiopia, with Egypt. These are empires that were existing back then, Aksum and what have you. So it is not known where this thing came from. But you can see the symptoms that were listed by some of the guys. This, as I said, killed a quarter of the population in Athens, including their leader, a renowned general who was called Pericles. But it was noticed that they suffered fever, they suffered redness of eyes, sore throat, bleeding. Somebody even mentioned bad breath, sneezing, loss of voice, coughing, vomiting, pustules and ulcers, diarrhea. So you can see, you can think of very many things. A lot of things can cause fever. A lot of things like even smallpox can cause pustules. Bleeding can come from different viruses. Diarrhea can come from different viruses. So for now, I would like to hear, what do you think caused the plague of Athens? If you can pick your phone and just type this into your browser, we'll be able to see what your thoughts are. There's no correct answer. So just tell me, of the many pathogens you know, which one do you think caused this plague of Athens? These are the symptoms, and this is the link. So just type P2. P2 is not what most of you are familiar with. P2.gg/K8E. Then just run that into your browser. It will bring you a text box. You can write there what you think caused this outbreak. So once everybody has typed this, I can go back. Actually, I can go back to the symptoms and just copy this so that we have people looking at the symptoms and thinking of what could have caused it. So this is the link, P2GG/K8E. And those are the symptoms. Let me see what people are writing here, if at all. You don't have to put your name or your email. You just submit. Of course, I don't mind if you put, but I think it's not necessary. You also don't have to share on Facebook, so make sure you click down there not to share. There are 2 submissions so far. Do I have 2 people? Maybe the rest are asleep. I can see some names here. Claire, Flora, Gillian, Hamza, Hope, Guta. There are 2 Hopes. Humphrey, Odenyi, Kigongo, Melva, Kidavasi, Makumi, Mlomi. Is it your area or your? Type something. Let's see. What you think could have caused it? And no one knows what caused this, so don't be afraid. No one knows what caused it. There are many theories, so there's no correct or wrong answer. Or there's Babu, is it Babu Owen or Babu Owino? So that is, now we have answers from several people. Thank you, keep them coming. There's no correct answer. So this is your opportunity to become a spy and try and figure out. As I said, this occurred when Athens was full of people who were scared and running from war, coming into Athens for protection because Athens was surrounded by water and their navy force was quite strong. It was stronger than Sparta and the other powers around it. So the people came into Athens for protection and the general retreated to protect Athens from within because Sparta and these other allies had very many, they had much more power in terms of army numbers. And with that, there was also trade. There were issues that the people suggested that the disease had come from Ethiopia because of trade. Ethiopia meaning Africa, not Ethiopia, Ethiopia. The part of Africa that was trading. This is mainly Sudan, Ethiopia, Eritrea, Algeria, Libya and what have you, Egypt as well. And also some thought it had come from Mesopotamia. This is Sumeria, the parts that are in Turkey, in Iran, in Iraq. And this is actually where the agrarian revolution, where the agricultural revolution started. So a lot of the viruses in this area had existed for a long time and they did not cause a lot of damage to the locals because the locals had been exposed to them. But then these areas were being controlled by the Greek. And so often the soldiers from Athens would be sent to these parts to control revolts and to assert their power, and then they would go back to Athens. That was a risk for infection. There are people who survived the infection, and those who survived the infection are the ones who took care of those who were sick. And people were dying a lot because there are mass graves that were found with hundreds of people, some of them looking like wells where people were just dumped into. So this was an outbreak that has reverberated or has reverberations throughout history. I was just giving you a story to see whether you will keep answering. So maybe we just open now and see what people have suggested. I want to share a window. And so what we said to do is p2 dot gg/k8e. Let me see if I can vote again. Vote something like what? And just say MSAKE so that we know it is not a disease. For your information, MSAKE is not an infection. Okay. So you can see yellow fever, Ebola, dirty water and crowding. I don't know what kind of infection that is. Bubonic plague, smallpox, measles, smallpox, typhoid, bubonic plague. So somebody thought it was caused by dirty water or crowding, but I don't know what that caused. It's an interesting thought, though, which is likely to have been the situation. We don't know how much it contributed. So thank you for that. Unfortunately, we still don't know what caused this outbreak. So people have suggested Ebola, people have suggested bubonic plague, people have suggested smallpox, people have suggested many other things. You can go and read around it and see whether you can crack the puzzle and get us evidence. I've mentioned already about SARS, and I don't want to spend much more time there. MERS-CoV was detected in 2012. MERS is Middle East Respiratory Syndrome coronavirus. Initially detected in older men who are interacting very closely with camels in the Middle East, and then spreading across parts of the world which interact closely with the Middle East. So from Saudi Arabia, people of course go to Egypt and the US, parts of Europe, China, and Vietnam and what have you. So all these are in Thailand. All these are examples of infection spreading across the world within a very short time because of the amount of travel that we managed to achieve over the last 100 years, which was not possible before. Previously, you could have a plague in one small village, the entire village could die, but well, it's unlikely that the entire village would die, but would really suffer like Athens did, but the rest of the world could be spared because there was limited interaction. I had not talked about the 4 points. This is Arizona, Utah, Colorado, and New Mexico. This is where the Hunter pulmonary syndrome started. And you can see within time, it spread to the rest of the country because of the interconnection, of course. Again, we've already described how people responded to that and how they were able to detect the cause. Chikungunya was interesting. I gave the example of chikungunya starting in East Africa, moving to the Indian Ocean islands. You can't see them here, but the Comoros, the Madagascar, the Reunion, Seychelles, all these were affected. And the virus later spread to India, then to the Pacific Islands, Europe, and of course to the US. This within 10 years, spreading across the world. Previously, only in East Africa, within 10 years, it was able to spread to the rest of the world. And you can see many of these famous holiday destinations and also a lot of people living in these areas because of population density. And then this also traveling across to the US, to Europe, which makes it easier to spread. Apparently, Russia doesn't get very many visitors as compared to Europe and the US, so sometimes they are spared from these issues. But of course, there are people from Europe going to Russia quite a bit as well. And of course, a bit from Africa. This is, again, just to re-emphasize the effect of travel. If you look at the map showing the planes that look like, I don't know what to call them, but they look like locusts, I think. If you look at Africa, and this is in March 2020, there's very little traffic. And it's not because of COVID. This is typically the story. If you look today, it's the same. If you compare with the same period, the existing traffic in Europe, in US, in Southeast Asia, in South America, you can see the density. And the same thing is reflected here, that initially, the number of cases of COVID-19, this is May 2020, is sort of directly related to the number of travelers. Because out of, for example, 100 people with COVID-19 traveling across anywhere in the world, only 2 will come to Africa, or 3 maximum. So, but once it comes in and it's established, then the local spread, community transmission, will be determined by what measures you take as a country. So within time, the effect of travel may be diluted. But the initial seeding rate, the seeding rate was determined by how many cases were coming in. And so that is important. That's why countries like Italy, which are very popular destinations for tourists from across the world, then would become culprits. The thing we need to learn is the process of vaccine development. It's quite lengthy. My point here is that you need to start early. You can't start when the infection has already occurred. And so with so much needs to be done before you can get to a vaccine. You start by understanding the virus, looking for things that in the virus that would be possible to make a vaccine. Then you test it in animals, small animals and big animals. Then you engineer the production process so that it's perfect. Some vaccines stop at this point. They cannot be produced consistently, good quality. Small scale clinical trials which test a few dozen people, usually tens, maybe 50, 60, will be looking at safety. Is this vaccine safe? Then large clinical trials, phase 2, which still small trials will maybe involve 100 people, 200, 300, a couple of hundreds to look at safety, but a bit of dosing and what have you. And then as you go into third or large-scale clinical trial, which takes thousands. You've seen some of the COVID-19 trials I've talked about 40,000. Typically, this is usually 5,000, 10,000. But some have actually, because of the urgency required, and the urgency we have had, getting 30, 40,000 people tested across different parts of the world. You can't just test in one part because they respond differently to, people respond differently to products such as this. So for you to be able to get to this large scale trial and then licensure, you need to have a way to do these other steps much faster. And if you don't have the tools, the personnel, the experience to do this, then you'll never get to produce a vaccine. So it's not just waking up one day and saying, give us a vaccine. It takes effort, and the effort begins with basic research. Even being as simple as culturing a virus, doing PCR, sequencing, generating antigens, and being able to inject animals and testing if it works. Luckily in Kenya, we have the Institute of Primate Research, which does this. And that is the end. I want to stop there. I know I've spoken quite a while. I would like to hear if you have any questions that you'd like us to address. Thank you very much for this opportunity. I'm glad to see MSAKE has grown. I remember we started these discussions when I was still in 4th or 5th year medical school with guys from Moi, and that time we were working under the umbrella of Federation of East African Medical Students Associations. And I'm glad MSAKE came into life, and it's been living since. Thank you very much.
Okay, thank you, Dr. Masika. We really enjoyed the session. I have learned a lot, and I expect that everyone has. We have seen things that we did not know, and I especially have realized that it's normal for Kenyans to assume that diseases and infections have one cause. For example, if you talk about malaria, if you ask the lay person, they'll tell you it's caused by mosquito, but it has a much broader aspect, and that you mentioned the One Health approach, and I think that is what has shown brightly from this session, viewing infections and diseases in a One Health view. And before I move to questions from the audience, I think you had asked why RNA viruses spread faster than DNA viruses. I think we should hear from the audience what they think before we move to the next part. So you can just raise your hand or post it in the chat box. Why do RNA viruses spread faster than DNA viruses? Let's see those who were attentive in their microbiology class. Okay, I'm not seeing anything in the chat box. I'm sure we're all attentive, and I think we know this. Most of us know this. Why do RNA viruses spread faster than DNA viruses? Okay, we have one answer in the chat box from Hamza. He says that the genetic material keeps changing, hence difficult to treat. Okay, Dr. Masika, you can respond to that.
Yes, that is true that RNA viruses, because they rely on RNA polymerase, which is not quite as error-free as, not really error-free, but it is more prone to errors as compared to the DNA polymerase. So more errors means more diversity. More diversity means a bigger chance for emergence of a new pathogen. There is also the element of crossing for DNA viruses, crossing the nuclear membrane, because every biological membrane crossed has some limitations. So maybe a virus will be able to infect somebody, but it may not be able to enter the nucleus for replication. So these are limitations that could affect DNA viruses.
Okay, thank you. So from there, we can move to the Q&A session. If you have any question, just raise your hand. We will unmute you and you can ask your question. Or if you're more comfortable with the chat box, just post your question there and it will be answered. Okay, as we wait for the questions, I think I would like to remind you all that today is the World AIDS Day, and HIV and AIDS is one of the emerging infections. And currently, there are about 36 million people living with with HIV, and I think this day was not made. It's there to mark the importance of being aware of HIV, its impact in people, what they go through mentally, physically, and especially economically, because the drugs required to combat the infection or to manage it are expensive. And so it's important to let these people know. They may be your family, your friends, just anyone basically. It's important to support them and know that even though they have this, we should not stigmatize them, but we should live with them knowing that this is something that can be managed and they can live as long as people without the virus do. So raise an awareness in any way that you know. It can be on your status, on social media, or if you can contribute money to organizations that help these people, just help in whatever way you can. And I think I had made a quiz that we will do at the end of the session to see how much you know about HIV and AIDS. So we can now go to the questions in the chat box. Thank you for this. We have the first one. I'd like to thank Dr. Lee for the informative presentation. I personally have gained a lot of new information with regards to microbiology. So that was not a question. It was a thanks to you, Dr.
Thank you. Thank you, anonymous attendee.
Okay, so for the second question, from the story of the plague of Athens, does that mean that the concept of immunity was documented for the first time in 430 BC?
I don't think so, because the history of disease is as long as the history of man. And for each disease, there were people who recovered and those who died. The infectious diseases began to create some gravity when people began living together in larger cities, and these or larger groups, and this was made by ability to produce food. If you can produce food, I think my internet is stable. Let me give it some time. If you can produce food and you can store it, it means you can feed more people. There are people to date. I saw a community in part of Tanzania that to date doesn't store food. I can't remember the name, because they just go and hunt and gather what they need and they eat. That community cannot sustain a large population growth. So with the agrarian revolution, or the revolution that brought in agriculture, where people could till land, then infections began to take toll. And this was around 12,000 years ago, or 10,000 years BC. And during that time, then infections began to come in. Some had been there for a long time, like herpes had been there for millions of years. But with this agrarian revolution, then people living together in cities, this was in Sumeria, parts of the Near East, and of course later the Nile Delta and in Egypt and what have you, where people were settling in large groups, then infections began to be documented. And even with that, people were recovering, and we could see that people recovered in some cases, like, well, for Athens, were charged with the responsibility or expected to take care of those who are sick because they were thought to be immune. And of course, there are more recent stories like the Jenna and smallpox and what have you. So I wouldn't say 430. I would say 430 is very recent in relative to people recognizing the role immunity plays in protecting against infections.
Okay, thank you very much. I hope you have been answered, anonymous attendee. If you have any more questions, just raise your hand. I will unmute you and you can ask them. We'll give around 5 more minutes for questions. So just raise your hand or post it in the Q&A box. Okay, as we wait, I think I can ask Dr. Masika, since
Yes.
you have mentioned that most of these emerging infections are because, let's say, because of mutations, pathogenic factors, environmental factors, can we to some extent say that we as humans are not learning as effectively as we should with some of these viruses and how they move from, let's say, from animals to humans? Do we play some part in that and hence contribute to the re-emerging of the viruses and the infections?
Yes, we play a big part. Remember, we also talked about human factors that contribute to this. And these are our interactions with other people through travel, through trade, our interactions with humans, through recreational activities, through hunting, through food, our hunt for food. For example, we have basically factories now for rearing chicken, where people are interacting with thousands of animals on a daily basis. So if there's an infection, it's very easy to get this across to humans. So as human beings, we have pressures, and due to these pressures of our daily lives. For example, if you take COVID, we've told people to stay indoors rather than going out, but somebody has to go look for food. Somebody has to go to deal with something that they feel is important. For some, they just feel like they have to go to the bar. For some, they feel like they have to attend a wedding. Some feel like they have to attend a funeral. All these are daily human pressures that sometimes overcome the fear of disease. I mean, if we were to be totally careful, we would never leave the house. That is the truth. But sometimes we take our adventure too far. And sometimes it's really just alignment, what I would call chance, that alignment of several factors together. That you went to hunt and you happened to come across the animal that had, the only animal that had a certain disease. And then it got you, and then you happened to go to people who are vulnerable. So all these are different things that must align. And I think this infection occurs so many times, but many times it's aborted. Because first of all, the infection will occur, but the person who has been infected is able to control the infection, or the virus is not able to actually go into the person's cells. And sometimes this person dies before they can infect others, or they are not able to infect others. And sometimes when it's a small area with very few people, then the infection is contained there. So there are many steps where this can occur. And yes, we play a role as human beings, and many times we do this without knowing.
Okay, thank you.
Karibu.
Okay, since I'm not seeing any more questions. Oh, okay, there is one more from Babu Owen. He's asking, is there a relationship between extreme hygiene at a young age and immunity?
Hygiene hypothesis. And I would like him to, Babu Owen or Babu Owen, to go and look at the hygiene hypothesis. That because of being exposed to several pathogens at birth or soon after birth, your immunity is trained to respond much faster. And this was the same theory that was being fronted for tuberculosis, BCG vaccine, and COVID, although that was later thrown out. But it is said that if for every for every pathogen that your immune system is exposed to, your immune system is trained so that you have some residual, better residual ability to respond even to general response to other infections. Although we know the most effective immune response is that one that is specific to a given pathogen. But when you have things like natural killer cells with trained immunity, that they are now better at responding to general antigenic stimuli or general pathogenic patterns, for example, then you you are standing a better chance of dealing with any new infection that comes your way. But again, this is called the hygiene theory. You can look it up and see whether you're convinced by the evidence presented that it's true or not. But I think to some extent it's true.
Okay, thank you. I hope Owen has been answered sufficiently. I think we can close the Q&A section for that. With that, sorry. So I think we have come to the end of our session. Thank you, Dr. Masika, for educating us. I'm sure we have all learned new things. We have learned a lot, and I'm sure we have been changed for the better. We have been made more passionate and enthusiastic about building that capacity to face situations before they actually occur, for example, with disease, and even as professionals when we finally complete our education. So thank you for joining us. We hope to have more collaborations with you in the future.
It's a pleasure to have you, and thank you for inviting me.
Karibu tena. So I think we can close with a quiz. I will share my screen. It is on quiz. Let me just text it in the chat box. myquiz.org. This is to raise awareness or to test your knowledge on HIV and AIDS in memory of the World AIDS Day. So just log into that. I'll share my screen. The quiz code is 609097. There are 15 questions in total, and each question is timed with 15 seconds to answer. The questions have different marks depending on the difficulty. And all the questions are related to HIV and AIDS. So let me just wait for you to log in. Just requires a nickname. You don't have to log in. Six 609097. I'll give us 1 more minute and I will start the quiz. Okay, I'm starting the quiz now. When should you start treatment? Let's see what people think. That's great. Everyone is of the idea that it should be as soon as possible, which is correct. How does HIV treatment work? Let's see what people think about that. Okay. Well, most of us agree that it reduces the amount of HIV. What does being undetectable mean? Great. See, we're all up to date with our HIV information and knowledge. Correct. Okay. I hope as you are answering, consider that the faster you answer, the more marks you have. Let's see who will respond fastest. People are fast. That's nice. As you can see, most of these questions are just basic and simple concepts that are important to know, especially for people who have to take the drugs, who have to live with this condition. And as friends and family, we should be informed so that in case they need our help or we need to take care of them, then we're doing the right thing and we're not putting their lives at more risk than they already were before. Okay. This one got everyone. It was a tricky one though. Which is a reason to visit your healthcare professional. What is AIDS? In the lead currently we have Sterling Forces, Claire and Hope. They've been in the lead for a while. Claire takes a turn there and goes ahead. Where did HIV originate? Chimpanzee, all of the above, mad cow disease, from hepatitis. Okay. It's good that most of us know documentation of this. Everyone with HIV gets AIDS. That is false. How does HIV become AIDS? This is one of the more technical questions involving the immune system, the cells. We are down to our last two. When was HIV first reported? Yeah, 1981. It was in LA. And the last question, what are T cells? This is fairly simple. Which is actually all of the above. The white blood cells, they scan for abnormalities and they coordinate immune responses. Great, so we have come to the end of the quiz. Thank you for participating. Let us look at our leaderboard. At the lead, we have Claire with 165 points, followed by Oceanic at position 2 with 155 points, and Hope with position 3 with 150 points. That's great. Thank you guys for participating. Kudos to everyone. I hope that you have learned something, not only from the session, but even from the quiz about HIV and the importance of raising awareness and being supportive of people living with this. So we have come to the end of our session. Thank you for coming. We have other sessions in the General Assembly. All will be virtual and all are free for the first time since MSAKE started having General Assemblies. We have one tomorrow. We hope to see you there. Have a nice evening, and everyone can leave at their own pleasure.